Hello, good morning everyone. My name is Nell Marie Pineda Solis and I'll discuss ketone bodies and ketogenesis. Ordinarily, most of acetyl coenzyme produced from fatty acid spiral is further processed through Krebs cycle. When oxacetate supplies are too low for all acetate coenzyme, to process through TCA cycle, ketogenesis takes place where excess acetyl coenzyme is converted to ketone bodies. Ketone bodies. Synthesis of ketone bodies from acetyl coenzyme is primarily in liver mitochondria. The three ketone bodies produced are acetoacetic acid, B-hydroxybutyric acid, and acetone. They are carried by blood to the muscles and tissues where they are converted back to acetoacetyl and then oxidized normally. During diabetes, however, the production of ketone bodies by the liver exceeds the ability of the muscles and tissue to oxidize them so that they are accumulate in the blood. Ketosis is the overall accumulation of ketone bodies in the blood. Ketonemia and in the urine is ketonuria. Ketogenesis. Ketogenesis is the metabolic pathway by which ketone bodies are synthesized from acetyl coenzyme A. Items to consider about this process prior to looking at the actual steps in four-step process are the first, the primary site for the process is living mitochondria. The second is the first ketone body to be produced in ketogenesis is acetoacetate. Third, some of the acetoacetate produced is next converted to B-hydroxybutyrate, the second ketone body. The fourth is acetoacetate and B-hydroxybutyrate synthesized by ketogenesis in the liver are released to the bloodstream where acetone, the third ketone body, is produced. Fifth is acetoacetate is somewhat unstable and can be spontaneously or enzymatically loose. Is carboxyl group to form acetone. Thus, the ketone body acetone is not actually a product of metabolic pathway ketogenesis. 6. The ketone body acetone present in the bloodstream is a volatile substance that is mainly excreted by exhalation. Its sweet odor is detectable in the breath or diabetic. The last is the amount of acetone present is usually small compared to the concentration of the two ketone bodies. The actual reaction steps in this process involve condensation, a second condensation, chain cleavage, and hydrogenation in that order. Biosynthesis of fatty acids. Not simply a reversal of the step of degradation of fatty acid. In biochemistry, lipogenesis is the conversion of fatty acids and glycerol into fats, or a metabolic process through which acetyl coenzyme is a converted triglyceride for storage in fat. Lipogenesis encompasses both fatty acids and triglyceride synthesis, with the latter being processed by which fatty acids are esterified to glycerol before being packaged into very low-density lipoprotein, or VLDL. Fatty acids are produced into cytoplasm of the cell by repeatedly adding 2 carbon units to acetyl coenzyme A. It occurs in most tissues, especially adipose tissue, liver, lactating mammary gland, and brain. Fatty acid degradation is the process in which fatty acids are broken down into their metabolites, in the end generating acetyl coenzyme A. The entry living organisms include bacteria and animals. The product of fatty acids oxidation are acetyl coenzyme A and reduced coenzyme FADRED and NADRED. The citrate malate shuttle system. Acetyl coenzyme A is starting to material for lipogenesis. Acetyl coenzyme A needed for lipogenesis is generated in mitochondria. Therefore, it must first be transported to the cytosol. Citrate malate transport system help transport acetyl-CoA to cytosol indirectly. Cytoplasmic acetyl-CoA is converted to malonyl-CoA in carbocitration, reaction that involves carbon dioxide and ATP. The reaction occurs only when cellular ATP levels are highly catalyzed by acetyl coenzyme A. Carboxylase complex which requires both Mn2 plus and biotin for its activity. ACP or acylcaryoprotein complex formation. First, all intermediate in the fatty acid synthesis are linked to carry proteins, ACP to SH. Second, ACP-SH can be regarded as giant coenzyme SH molecule. Chain elongation. Chain elongation is an anaerobic open culture biotechnological process that converts volatile fatty acids or VFAS and the electron donor into more valuable medium chain fatty acids or MC-Fas. Microsomal fatty acids elongation represent the major pathway for determining the chain length of saturated, monosaturated, and polyunsaturated fatty acids in cellular lipids. The overall reaction for fatty acid elongation involves 4 enzymes and utilize malonyl coenzyme, NAPH, and fatty acyl-CoA and substrate. 4 reactions constitute the steps of chain elongation process. Step 1. Condensation. Step 2. Hydrogenation. Step 3. Hydration. Lastly, step 4. Hydrogenation. And that's all about ketone bodies and ketogenesis and biosynthesis of fatty acids lipogenesis. Thank you.